This is the second of 3 mini lectures that will address these 3 objectives with regards to the initiation and function of the adaptive immune response. And this mini lecture will focus on T-cell effector functions. Here's an outline of some of the key subjects we'll talk about today. We will discuss the function of Th1, Th2, Th17 Treg, and cytotoxic T lymphocytes. And lastly, we'll end with a discussion of memory T cells and their role in protection from disease. So it's important to note that T cells migrate to inflamed tissues using very similar mechanisms that are utilized by neutrophils and monocytes. And previously we talked about the role of ICAMs and selectins and integrins like LFA1 in recruiting neutrophils to sites of inflammation. The mechanisms recruiting effector T cells are very similar, and so we're not going to review those in this lecture today, but it may be a good idea to go back and review that material from the innate immunity lectures so you have a good understanding of how these T cells to get into tissues. The other thing that's important to note is that the different effector subsets are each recruited by a unique array of chemokines. So chemokines really control the flavor of the inflammatory response that occurs within tissues. Now, when Th1 cells migrate out of the capillaries into the tissue and to the site of infection, they meet up with macrophages. And if you'll recall, macrophages are one of our key professional antigen-presenting cells that express MHC class II. Macrophages have been busy phagocytosing whatever pathogen is present in their environment and attempting to degrade that pathogen within their phagolysosome. And so it's able to present some of these peptides on MHC class II molecules to Th1 cells that migrate to those tissues. When this happens, this allows Th1 cells to provide 2 key mechanisms of help to those macrophages. The first is the engagement of the CD40 co-stimulatory pathway. T cells express CD40 ligand. When this binds CD40 on the surface of macrophages, it enhances their activity. The second of those key functions that Th1 cells mediate is the secretion of interferon gamma. And interferon gamma binds to the interferon gamma receptor that's expressed on macrophages and also induces the activation of these macrophages. And so you have a good macrophage. This macrophage can mediate antimicrobial effector functions and can phagocytose and can destroy this pathogen within the phagolysosome. But when this macrophage gets activated by Th1 cells, it becomes a great macrophage, highly activated, much more efficient at destroying bacteria within the phagolysosome. So what is different about these activated macrophages? Well, they have enhanced phagocytic activity. They increase the expression of NADPH and the production of nitric oxide within the phagolysosome, and so they have enhanced phagolysosome activity. They increase MHC class II expression, so they have enhanced antigen presentation. They increase their production of inflammatory cytokines like interleukin-1, interleukin-6, and tumor necrosis factor, and really become effective at eliminating all intracellular pathogens. And by intracellular, I mean pretty much anything, because virtually all pathogens can be phagocytosed and enter into this phagolysosome pathway. And so Th1 responses play a really critical role in antibacterial defense, antiviral defense, antifungal defense, and even for some single-celled parasites. Now, the Th2 response is more important for promoting protection from helminths. And you'll learn more about helminths later in this course, but helminths are worms that form parasitic relationships with humans. And in the industrialized world, helminth infections are not nearly as prevalent as they are in other parts of the world, or as they have been historically in the interaction with human populations. But historically, the Th2 response has been incredibly important for protecting from these types of infections. So what do Th2 cells do? Well, they secrete 3 cytokines, IL-5, IL-4, and IL-13. IL-5 induces the activation of eosinophils and mast cells and can induce their degranulation. And the contents of these granules have really potent anti-helminth activities. On the other hand, IL-4 and IL-13, when this Th2 cell is interacting with a macrophage, can induce this macrophage to change its activation status from a classically activated or M1 macrophage to an alternatively activated, or M2 macrophage. And this alternatively activated macrophage starts to secrete cytokines that one would consider more immunosuppressive, such as interleukin 10. And it's really important in wound healing and in resolution of inflammatory responses. And so many macrophages in the late stages of inflammation, as inflammation becomes resolved, are actually of this type, and generally suppress the inflammatory response and tune down inflammation. Th17 cells secrete two key cytokines, interleukin 17 and interleukin 22. Interleukin 17 specializes in inducing the mobilization and recruitment of neutrophils. And these neutrophils, of course, have really important functions in eliminating extracellular pathogens such as extracellular bacteria. Interleukin 22 acts on epithelial cells, often at the gut barrier or in other mucosal locations, and induces increased expression of mucins, defensins, lysozymes, all sorts of things that act in the lumen of these mucosal barriers to prevent colonization of bacteria. And as one might expect, Th17 cells are abundant in mucosa-associated tissues. Lastly, T-regulatory T cells suppress activation of antigen-presenting cells. So this slide is a little busy, but it's just meant to indicate that regulatory T cells secrete immunosuppressive cytokines such as interleukin 10 and transforming growth factor beta that suppress the activation of dendritic cells, they suppress the activation of macrophages, and they suppress the activity of T helper cells. Additionally, regulatory T cells, when they interact directly with macrophages and dendritic cells, induce a lot of negative signals that reverse their activation and cause them to become tolerogenic. And so dendritic cells, for example, that have interacted with these regulatory T cells will now specialize in suppressing T helper responses instead of activating T helper responses. And so Tregs can initiate kind of a cycle of immune suppression. So both contact and cytokine-dependent mechanisms of suppression are seen. It's important to note that there's two main sources of Tregs. The first is during thymic selection, Tregs can arise and are important for enforcing immune tolerance to self-antigens. And second, during immune responses, Tregs can be induced. And these regulatory T cells are thought to prevent excessive tissue damage mediated by these immune responses. So again, these play an important physiological function in preventing T cell mediated immunopathology. Lastly, let's talk about cytotoxic T lymphocytes. And cytotoxic T lymphocytes specialize in mediating cytolysis of virus-infected cells. So here we have a CTL that is interacting with a virus-infected target cell. And this CTL forms a cytoplasmic bridge with its target. And once it forms that bridge, it starts to secrete granules that allow it to kill this target cell. These granules contain an enzyme called perforin that forms pores in the membrane of the target cell, and an enzyme called granzyme B, which once it enters into the cytoplasm through those pores induces apoptosis of the target cell. And this is the primary mechanism by which cytotoxic T lymphocytes kill their targets. There is an alternative mechanism by which cytotoxic T lymphocytes can kill their targets, and that is because they can be induced to express an apoptotic ligand called Fas ligand. And Fas ligand can bind to Fas on the surface of these infected cells, and this in turn induces an intracellular signal into the cell that induces apoptosis of the target cell. So both of these pathways result in apoptosis. Again, the primary pathway is this granule-mediated killing. I just did want to make you aware that there are alternative mechanisms by which cytotoxic T lymphocytes can induce apoptosis in their targets. So this graph is meant to indicate the kinetics of a T cell response in vivo. So we've learned about the activation of T cells that get primed and then start to undergo clonal expansion and acquire effector functions. And this can be in response to a virus or a bacteria that causes an acute infection that proliferates, and then as this adaptive response expands, this pathogen is controlled and cleared. This expansion of T cells from a very, very few precursors, like I mentioned, is 100,000-fold expansion in some cases and can mediate clearance of the pathogen. Once the pathogen is gone, most of these effector T cells die off. And this is a period called contraction. And in this period of contraction, 90 to 95% of the effector T cells die, leaving behind a long-lived population of memory T cells that can respond to secondary challenge. One of the benefits of these memory T cells is that they are present at higher frequencies than they are at the naive state. And so you maintain a memory of your infectious history. You maintain cells at higher frequency that target antigens that you've seen before. Secondly, these memory T cells can be very long-lived and have the capacity for self-renewal, up to a lifetime in some cases. And I think in cases of smallpox vaccination, memory T cells can be detected circulating up to 75 years after the vaccine. And lastly, these memory T cells, if they do see their antigen again, rapidly reacquire their effector functions and become very efficient mediators of pathogen control. And so memory T cells form an important part of immunological memory. Okay, so again, here's a summary of this mini lecture. Please take a moment to go through these points and review the material that I presented today.